Hello everyone. In previous sessions under molecular basis of inheritance, I have covered introductory part and the double helix structure along with chemical composition of DNA molecule. In today's session, we are learning different experiments carried by number of scientists, out of which 3 experiments are included in the syllabus to search genetic nature of DNA molecule. It was 1928. Frederick Griffit was working on pathogenicity of a bacteria Streptococcus pneumoniae. This carry 1 mark weightage. Name of the person? Frederick Griffit. The year 1928. Name of the experiment? Griffith's transformation experiment to prove genetic nature of DNA. The bacteria used for experimentation? Streptococcus pneumoniae. A bacteria responsible for pneumonia, a respiratory disorder in human population. When he cultured these bacteria on a culturing plate, now cultured means what? He took bacteria in the laboratory, provided them a nutrient medium so that they can grow rapidly by binary fission. When he observed the cultured plate of Streptococcus pneumoniae, he saw 2 strains. Some bacteria were with shiny surfaces, with capsule, and some bacteria are without shiny surface, that is without capsule. He named bacteria without capsule as R bacteria, R specifying rough bacteria. He specified capsulated with shiny surfaces bacteria as S bacteria, stands for smooth bacteria. When Griffith observed cultured plate of Streptococcus pneumoniae, he got 2 strains R, S, rough and smooth. He was under confusion. Now, which bacteria is causing pneumonia? So, he tried the experiment by taking mice. Mice are the white albino rats, which are oftenly used to learn the effects of chemicals, effects of certain processes at the molecular level. I'll explain one by one what Griffith did. You have to remember like a story, then only it goes easy to write the answer. He took mice. He injected R-type bacteria in it. He took another group of mice. He injected S-type of bacteria. He started observing. Now there are 2 groups of mice. Mice injected with R-type. R is rough, non-capsulated. And second group of mice injected with S-type, means injected with smooth capsulated bacteria. After some days he saw mice injected with S-type died, but mice injected with R-type survived. So he concluded that S-type capsulated bacteria causes pneumonia. They are pathogenic. R-type bacteria are non-pathogenic. He was Griffith, not like us. He was curious about whether we can kill pathogenicity of S-type or not. So what he did, he cultured S-type, that is smooth bacteria which were pathogenic. He cultured them on a large scale and he treated them with the chemicals. For this he used the word attenuated pathogens. Heat killed pathogens, paralyzed pathogens. So Griffith in 3rd attempt took S bacteria from the culture medium, treated with the high temperature and attenuated them. Means he half killed them, paralyzed them. He took third group of mice and injected heat-killed, attenuated, paralyzed S bacteria in it. And he observed third group of mice injected with attenuated smooth bacteria were absolutely normal. So he concluded that we can kill pathogenicity of bacteria by treating them with certain grades of temperature. He made fourth experiment now. He took smooth bacteria, attenuated them, paralyzed them, and normal R bacteria, made a mixture. And this mixture he injected in the fourth group of mice. After a few days he saw fourth group of mice developed pneumonia and died. When he saw blood samples of dead mice, in blood samples he saw attenuated bacteria, he saw smooth bacteria, not rough bacteria. For this he called transformation. Means normal R-type bacteria, when it is mixed with attenuated smooth bacteria and injected in the host, that is in the mice, R developed a capsule, become smooth. They developed a pathogenicity, and by which mice got infected by pneumonia, resulted into death. But he was not knowing what exactly happened, what exactly the phenomena by which R bacteria converted into S. Non-pathogenic become pathogenic, non-capsulated become capsulated, non-virulent becomes virulent. He just said that there is a transformation in bacteria. So there is a chemical responsible for this transformation. This is called Griffith's experiment to prove genetic nature of DNA. But here he was not aware of DNA molecule. He just showed there is a transformation. As a history part of this experiment, before Griffith, Lederberg and Tatum conducted experiment on E. coli. They cultured Escherichia coli and they saw in the culture some are with flagellae, some are without flagellae. He saw flagellated and non-flagellated started conjugating with each other. Means they started pairing with each other. And after some time they separated and non-flagellated developed a flagella. For this they called conjugation. It is a type of sexual reproduction in bacteria. Later they analyzed that the flagellation, appearance of flagella, is a morphological trait in E. coli which is controlled by a gene, and gene is a part of DNA, right? So why they come together? Flagellated had that gene which is transferred to non-flagellated. This transfer of gene from flagellated to non-flagellated they call conjugation, and because of this transfer non-flagellated become flagellated. In this session you please remember the Griffith's experiment to prove genetic nature of DNA, but he was not concluded that DNA is responsible for transformation. With this I will conclude my today's session. Thank you very much.